All right, so let's start with any questions y'all have. Anything at all? Anybody online questions? We don't have too many people here today. All right, my computer's saying that we have unstable internet. Through HD meetings that I wasn't aware of that we could do. So over the 2-week break that we're going to have, I'm going to think about some things that we can do to work in small groups so that you're not just listening to me talk for an hour every day. So I'm going to try and change the structure of this course for if you've seen other classes do it, give me ideas and I'll roll with them. Okay. So let's go ahead and go into the slides for today. All right, so I want to start by just talking about something that Sabrina mentioned yesterday. So we were talking about the proteobacteria as a group of gram-negative organisms. And the book says that only gram-positive organisms are going to produce endospores. But I found a paper, this paper right here, as I was preparing yesterday, that said that some of the delta proteobacteria included within a genus called Myxobacteria can produce this multicellular-like state. Remember we talked about that? And they can produce spores. These are gram-negative organisms. So that breaks the rule that we said no gram-negatives produce endospores. So then, if there's an exception to the rule, I expect there to be another one. So I went digging around yesterday, and I found another organism that's gram-negative that can produce endospores. And this paper is from 2013. Your book is from 2016. So our book isn't always right. Always be skeptical. Always think about what you're learning. All right, so let's go through viruses today. This is going to help us prepare for the exam. Keep in mind we're doing review tomorrow. This should also help give you a little jump start for your paper that you're going to write too. So talking about viruses, the first virus that was discovered caused tobacco mosaic disease. So this is in the late 1800s. So we already have a germ theory of disease. Who proposed the germ theory of disease?
Pasteur.
There you go, Louis Pasteur. Pasteur, germ theory of disease. I think it was 1864. All right, so late 1800s, this group discovered the tobacco mosaic virus. So let's get our little pointer here. So you can see this tobacco leaf. It has some discoloration and sort of a mosaic pattern. Think mosaic like you put tiles together, little broken pieces of glass. So amongst the green, we see some yellow, some brown spotting. And so what they did is they tried to filter it. So they ground up the leaves and then passed it through a filter. So what do you think they were looking for to be stuck in the filter? Probably looking for bacteria, right? But what they got in their filtrate was this contagious living fluid. But that didn't fit with the germ theory. So again, we don't have contagious air. Do you all remember what the name for contagious air was? Miasma, right? So it's no, there are infectious particles, infectious agents in the air. We don't just have fluid that is infectious. There's something small in it. And that is how we discovered viruses. So that was late 1800s, go up a handful of years to 1901. And Walter Reed was studying yellow fever. This is also caused by a virus. So now we know viruses can cause disease in plants and humans. So we discover a virus, and then as soon as we discover it, we find that they are infecting everything that we can look for. 1915, a group is studying bacteriophages. We'll talk about them today. A bacteriophage is a virus that infects bacteria. So now we have viruses infecting plants, humans, and bacteria. 1930s, it was claimed that viruses are not alive, so they were able to crystallize a virus. They found it doesn't have cytoplasm, it doesn't have organelles, it is protein encapsulating a nucleic acid. So this is less than 100 years ago, so relatively new. This is like 2-3 generations old science. So then once we learned about what a virus was in the 30s, in the 40s, there was a culture technique used. Your book shows an image of this towards the end of the chapter, where you could take a fertilized chicken egg and inject a virus into it, and then that can grow more virus. So the machinery of the egg is used to produce more viral particles. And then this figure here just shows how small a virus is relative to our cells. Notice that some of them are huge, about the size of a bacterial cell. If you are the size of a bacterial cell, you're not going to infect it and get in it, right? So notice just how small a bacteriophage is relative to something like a Pandora virus. So there's a lot of diversity in the size as well as morphology of viruses. But they're all going to be smaller than our cells. So relatively, they are very small, very tiny. They are always going to be obligate intracellular, and the book calls them parasites. I'm going to call them a parasitic entity, just so that we can avoid even hesitation, or just, I don't want you to have any sort of a thought about these being alive. They're not alive, so I'm going to call it a biological entity or a particle, as opposed to just a straight up parasite, which we typically refer to as like a eukaryote, like a worm. But just so you all are aware, a parasite is anything that's going to extract nutrients from us without giving us something. So it's a pathogen. So if they require our cells for their replication, they're going to do that because they can't replicate on their own. And at the end of today, we'll talk about some theories about how they arose in the first place. But all of them are going to need a host cell to replicate. They'll replicate in a couple of different ways. We'll talk about the lytic and the lysogenic cycles. But again, just keep in mind, a virus is a genome consisting of RNA or DNA. Not both, it's one or the other, which I think is interesting. It can be double-stranded or single-stranded, but it's going to be one of these two nucleic acids. This slide contains a handful of definitions about parts of a virus. So the genome, again, is a nucleic acid, DNA or RNA, and then it's surrounded by a capsid. So the capsid is this protein coat, and it's made up of individual, I guess we could call this like a monomer component of it, which is the capsomere. So each of these little ball shapes in this yellow part is going to be a capsomere. Now, the book says that these spontaneously arrange around a nucleic acid. I'm assuming that has to do with some sort of hydrophilic, hydrophobic interactions in their environment, but it doesn't go into detail about that. So on the outside of the capsid, some viruses are going to have what's called a spike protein, and this is going to help the virus attach to a host cell. You need to attach before you can get inside. So attachment is always necessary for an infection to take place. So if we look over here in part A of this figure 13.5, this non-enveloped form we can describe as a nucleocapsid because it contains a nucleic acid surrounded by a capsid. It lacks an envelope. So we could call this a non-enveloped form of a virus, or just strictly a nucleocapsid, or a naked virus. An enveloped virus is going to be that nucleocapsid with an outer surrounding of lipids and proteins. And so that's shown here in part B of 13.5. Do you all remember in your reading where this envelope is derived from? Yes, this is going to be derived from the host cell through a process called budding. And then we'll also see a figure whenever an enveloped virus enters into a host cell, its envelope integrates into the host cell membrane as it enters in the cell. And if you are an enveloped virus, the region between the capsid and the envelope is called the matrix. And these are all just definitions. So, does a naked virus have a matrix? No, it does not, because it does not have an envelope. And so, if a viral particle is completely assembled outside of a host cell, we will call it a virion. So, after, let's say, the lytic cycle, so a cell ruptures through the process of lysis, all those viral particles are now called virions as they enter into out of a host cell. So we can talk about shapes. When I say shape, I'm referring to the capsid, so the protein coating around the nucleic acid. There are three types of symmetry. Helical is a cylinder that's going to wrap around the nucleic acid. Icosahedral is a 20-sided capsid. Pretty cool. And then we can have a combination. We're going to call this a complex symmetry. So something like a bacteriophage has a 20-sided capsid attached to a cylindrical helix capsule as well. Capsid, pardon me. So you're a cylinder, 20-sided, or both. More definitions. So we have a host range. That's going to refer to which organisms a virus is able to infect. Can you all think of something that's going to be necessary for infection to take place? So what's going to make something able to infect a given cell type? What do we have to do? So we're going to have to replicate. Before we replicate, what do we have to do? Yeah, we have to attach, right. So if certain organisms lack a receptor that a virus needs to attach, it can't enter into that type of organism. So that's the compatibility between a virus and its host, is can I attach? A virus might not be able to attach, but then it can mutate, and then maybe it can be able to attach after that. So that's the host range. So that's going to depend on the most exterior structure because, again, we need to attach to infect. If you are not enveloped, that attachment is going to be on your capsid protein, so the spike. If you are enveloped, it's going to require some sort of a ligand on your envelope to attach to a host cell. So it's your outside. And then this figure here shows just that. So if the influenza virus attaches to the outside of a host cell at an area called sialic acid, if you remove the sialic acid, The virus can attach. If it can attach, it can't get in. So we can experimentally show how this happens. Certain viruses like certain cell types better than others. We would call that cell tropism. Or if they like a certain tissue better than others, we would call that tissue tropism. So for example, the book mentions herpes viruses residing in the trigeminal nerve. So that would be a cell tropism of a neuron, as opposed to something like an epithelial cell. All right, this is worth studying for the exam on Thursday. Just how are we going to organize things? I don't expect you to memorize the names of these organizations. I want you to know generally what exists as far as the diversity of viruses. I want you to know that DNA viruses can be single or double-stranded, enveloped or non-enveloped, and the same goes with RNA viruses. I want you to know that. Within the RNA viruses, I want you to understand the difference between a positive and a negative strand. And I also want you to know what a retrovirus is. That's down here at the bottom. So from this figure, you should know diversity includes single double-stranded DNA or RNA as far as the nucleic acid component of a virus. Anybody remember what a positive strand RNA virus is? Yes. So you can think of a positive RNA virus as it's similar to our messenger RNA. So this is injected into a host cell cytoplasm. It's going to go directly to a ribosome. And we have replication occurring at the ribosome. So the way this is going to happen, the RNA viruses include in their code an RNA, follow me here, RNA-dependent RNA polymerase. We make RNA from DNA. So that would be DNA-dependent RNA polymerase, right? Viruses can encode their own nucleic acid transcription machinery. So they can replicate their own, I don't want to call it a chromosome, but nucleic acid. And then the ribosomes are going to make the capsid proteins that go around it. And you don't need to know the name of this committee. Okay, a DNA virus can be, again, single or double-stranded, but it is a DNA virus, and if it's going to replicate within a host cell, that's going to be in the nucleus, because that's where our DNA polymerase is located. The nucleus contains the chromosomes, the exception to this rule, and I do want you to know this, are the pox viruses, because they encode their own DNA polymerase. So they don't need to go into our nucleus to replicate. We mentioned that RNA viruses are going to be replicating in the cytoplasm because, again, they are already in their transcribed form. They just need to translate at the site of the ribosome. And ribosomes are going to be in the cytoplasm. And then I said I want you to know retroviruses. So HIV is an example of a retrovirus. This is an RNA virus that is going to go into a double-stranded DNA form. It incorporates itself into the host genome, so that we can create more RNA. So it goes RNA to DNA to produce more RNA of the cell. And I'll show you a figure of that. So this is the human immunodeficiency virus, HIV. So it's going to enter into a cell. So its nucleic acid is RNA. Reverse transcriptase is an enzyme that's going to create a double-stranded DNA from a single-stranded RNA template. That double-stranded DNA integrates into the chromosome. So that means as the cell divides, we're going to have that virus in the new daughter cells. So then as our machinery in the nucleus transcribes proteins, transcribes mRNA, we can transcribe that virus out, and then we have new viral particles leaving cells. This is a retrovirus. So if it integrates itself into the genome, it can stay hidden from the immune system. That's an adaptive strategy of a virus. Pretty sophisticated. We're going to see these processes over and over again today, so I think they're worth committing to memory. So again, you have to attach before you can do anything as a virus. After attachment, then you want to get into a cell. So you're going to find a cell, attach to it, get in it. And then you're either going to go into a latent or kind of a dormant phase where you lay low for a little while, or you'll just go ahead and start producing more of yourself. So biosynthesis, making your proteins, replicating your nucleic acids. After you've created your parts, then you will assemble them and then eventually release so that you can go do it all again. All right, so now we're going to talk about viruses that infect bacteria, and then we'll go into animal and human viruses. So what we're talking about now, bacteriophages. These are viruses that only infect bacteria. Now, we can be infected with bacteria that are infected with viruses, but a bacteriophage will not infect a host cell. So now let's go share, PowerPoint.
There we go.
Can you see the PowerPoint or my camera now?
PowerPoint.
Okay, great. Okay, back at it. All right, so the Lytic cycle, this is where cells are going to lyse, or you think of this as they're going to blow up. They're going to rupture. And that's going to be the end product, step 5, where we release new particles. can do. Okay. So we're going to send our nucleic acid in the cell, create more viral particles, they'll mature, they'll assemble, and then through lysis, they'll rupture. And this